Welcome to today's Cell and Gene Therapy lecture. We are organizing this from the side of Miltenyi Biotec, a biotech company strongly engaged in cell and gene therapies. I am Apel and I'm accompanied by my colleagues Philipp and Martina. First of all, I'm presenting you a short disclaimer on our work, on our background. And the agenda is as follows. I'm giving you an introduction about Miltenyi on Miltenyi Biotec. Afterwards, I'm talking a little bit about cell and gene therapies, current applications and opportunities, followed by challenges and future prospects. And afterwards, Martina and Philipp will take over with the second part and give you more details on the manufacturing of CAR T cells. Miltenyi Biotec has a mission. It is on innovative cell and gene therapies. We want to cure chronic diseases like cancer and autoimmune diseases, as well as genetic diseases. Some fast facts about Miltenyi. The company was founded in 1989 and meanwhile has almost 5,000 employees, and 25% of them are working in R&D. And we have a huge amount of products, meaning almost 20,000 products that Miltenyi sells. With our technologies, more than 150,000 patients have already been treated, and every year 10,000 further patients are treated with our products. We are present globally in 24 countries with direct sales and will reach 1 billion sales next year. Some words to our core technology. It is called magnetic cell sorting, the MACS technology. And the core of the magnetic cell sorting is when you have a mixture of cells, which is illustrated here, and you want to isolate a specific cell type from this mixture of cells, which is this purple cell, then we label these cells with magnetic antibody nanoparticles that are indicated here by this Y-shaped molecule, and the nanoparticle is the bubble on top. And when we put this mixture of cells into magnetic fields and transfer this cell mixture over a column in this magnetic field, you can the labeled cells are retained on the column, and the non-labeled cells they pass the column and can be isolated. On the other hand, when you take away the magnetic field, you can elute the retained cells, the target cells, and isolate them as well. Now, coming to current applications and opportunities. The concept of personalized cell and gene therapies is illustrated in this slide. You isolate cells from the patient, meaning you isolate own cells, autologous cells like immune cells or stem cells. Then you engineer them and you create a modified cell product, which is indicated here by these different colored cells. And you also expand usually the cells. And then you transfuse them back into the patients where they do their job and cure the patients cure the patient's disease like cancer or some autoimmune diseases, for instance. And all this can be done with our CliniMACS Prodigy in an automated way. The Prodigy is an instrument that handles the entire circle that I have shown here in an automated closed system. What is the principle of CAR T cell therapy? We are redirecting the T cells with synthetic antigen receptors that are called chimeric antigen receptors. And the concept is shown on the slide down here. Here you have the T cell that has a chimeric antigen receptor, and on top you have the tumor cell that should be attacked. The CAR molecule recognizes the antigen expressed by the tumor cell, and if this recognition happens, then a signal is given by the T cell to destroy or kill the tumor cell. Why are we focusing on CAR T cells in this lecture? Because CAR T cells represent the majority of all cell types that are used for cell and gene therapies. They represent approximately 49% of all cells. Other cells are stem cells, for instance, dendritic cells, CAR and K cells, meaning natural killer cells, or other stem cell types and other immune types. The clinical focus, of course, is oncology, also because of CAR T cells and immune cells. Oncology is 74% of all cell and gene therapies that are done. And of course, we have some other indications, but they are much less represented in this overview. Coming to challenges and future prospects. One of the challenges is illustrated here. It is the global imbalance of clinical trials. You see that most of the clinical cell and gene therapy trials are happening in the United States and in China. Europe just plays a minor role in this field. There are only a few percentage of clinical trials happening in Europe, and in comparison to the US and China. And another challenge are the high price tags of those of cell and gene therapy products. Here on the bottom, you see the price tags for Kymriah and Yescarta. These were the first two CAR T cell products that were approved in the US. Already in 2017, and the price tag of those therapies is between 370,000 up to almost 500,000 euro per cell product, meaning that every patient who gets this treatment has this price tags on these costs. There are even more expensive cell and gene therapy products, as given by Zolgensma, which has a price tag of more than 2 million euro, and this is not the maximum. Meanwhile, since 2021, there are also other products approved that are even more expensive. On the other hand, despite these challenges, CAR T cell therapies bear many opportunities. It is a one-time injection that can cure a patient. It is curative in patient even at the latest stage of cancer. It is a living drug, meaning it has different properties, better properties than conventional drugs. It is flexible to new applications, and also suitable for personalization, meaning that every patient gets his personalized cell product. The challenges, on the other hand, I already mentioned the high cost or the high price tags. It is effective so far in only a few rare cancer types, meaning cancers of the blood. Solid tumors are difficult to treat. There is these treatments, of course, have side effects. We have the cytokine release syndromes, for instance, is one of the side effects that is very common, but it can be handled meanwhile. And the manufacturing that we are going to talk about today in more detail is very complex, as you will see, and therefore the access for patients is limited. And also, the complex manufacturing and gene therapies in general have complex regulatory hurdles that physicians have to deal with, and also researchers when they want to perform a clinical trial. Miltenyi Biotec has founded a pharmaceutical branch in 2020, which is called the Miltenyi Biomedicine. And the focus of this company is the facilitating of clinical CAR T cell trials in Germany, Europe, and worldwide. And we are working on many serious indications like cancers, and we try to develop innovative therapies for treatment. In this Samtocaptagin autoleucel trial, which is a DALI trial, we use a target that is directed against, we use a CAR that is directed against two targets in parallel, which is, therefore we have a dual target approach. And we are already in a pivotal trial, meaning that we are starting or finalizing our clinical trial next year, and hope that we will get an approval to treat a rare type of cancer cells. Furthermore, another new development is that we treat autoimmune diseases with CAR T cells right now with the CD19 CAR T cell, and I will come to this in a moment and explain a little bit more on this. And also solid tumors, which have been difficult to treat. We have also started a phase 2 trial, which is called GD2-IL18, where we want to apply a new target molecule in combination with an interleukin to treat solid tumors like neuroblastoma or breast cancer. Coming to our main or most important trial, which is this samtocaptagene trial, which is targeting an aggressive B-cell lymphoma, where there are only pure therapeutic options for patients right now. What is the principle of dual targeting? Tumor cells express, next to a variety of different target antigens, they express CD19 and CD20 in these lymphoma patients. And we are with our CAR construct, we are targeting both of these constructs. We have two binding sites in the CAR construct against CD19 and CD20, and thereby we want to prevent tumor escape. Because tumor cells sometimes lose one of those antigens, and then they can escape the response of the CAR T cells. But if you have two binders, then you increase the chance that the CAR T cell can attack the tumor cell and destroy it. We have already performed first phase 1 clinical trial, which is illustrated here. The results were very promising because 75% of the patients responded to the CAR T cells, and 5 of 12 patients had an ongoing response that was lasting longer than 1 year. In addition, there are new areas of application for CAR T cells. We are trying to expand the CAR T cell therapies now to autoimmune diseases. And researchers from Erlangen that are shown here in this photo, they had the idea that you can target autoimmune diseases using a CD19 CAR T cell product. Why CD19? Because CD19 is expressed on B cells, and B cells secrete antibodies that cause the autoimmune disease. And these antibodies are autoreactive and destroy or attack own tissue. And they wanted, with these CAR T cells, they destroy the origin of those disease and can eliminate the B cells that are causing the disease. And they have already treated 14 young patients with advanced disease, and have already reached very promising results that could be published in Nature 2 years ago. Another new technology I just wanted to mention is in vivo gene delivery. That this is really the future that we will see in 10 years' time. Presently, the CAR T cells, they are generated ex vivo. With the CliniMACS Prodigy that Philipp and Martina are going to explain in the next minutes, usually the cells are generated outside the body ex vivo and in a very complex manufacturing process. What the future will bring is that you can use lentiviral vectors that you need for the manipulation of the CAR T cells You can inject those vectors directly into the body where they transform the T cells in the patient, and then you generate CAR T cells in the patient with site-specific or with cell-specific vector constructs. And this will save time and be much faster for the patient. You have no complex ex vivo off-the-shelf manufacturing. The patient burden will be lower because you don't need a leukapheresis or lymphodepletion. Toxicity hopefully will be reduced, and also we expect a better efficacy for the patients. Last but not least, from my side, I'm part of the grant team at Miltenyi that is shown here. And if you want to have more information, you can join our website or follow us on LinkedIn. That's it from my side, and I'm going to hand over to Martina right now, who will give you more insights on the CliniMACS Prodigy.
Thank you, Michael. Now I would like to introduce to you our CliniMACS Prodigy system. Beforehand, we will have a look at the CAR T-cell manufacturing process. And as Michael already mentioned, it is a multi-step process, with a lot of single steps that bear also unintended risk and cost if it's done manually. So starting from the sample that needs to be washed, where the T cells needs to be selected, activated, transduced, expanded, up to the harvest and the filling of the final product. That can be more than 20 transfers and manual handling steps, and you might need many devices that give windows for variability and errors. Furthermore, if you think about open steps, these require higher clean room classes and higher requirements, and these manual protocols also need highly skilled staff, so highly trained personnel. Furthermore, if you think about different devices in your process, these require different protocols. You need multiple IQ/OQs and maybe also service contracts. Furthermore, you have some kind of disconnected data flow, and the recording is not so easy. So an automation of this process is highly desirable, especially to get a more scalable, robust, and reliable process that will enhance the product quality. So the CliniMACS Prodigy instrument is the so-called integrated cell processing unit. And you see on this picture here the most important feature of the device, which are here the touch screen, where you can choose the right software for the kind of cells you would like to manipulate. You have here in the middle the magnet unit, where the cell separation takes place. You have here a peristaltic pump that is moving fluidics within a closed tubing set. And you have here a crucial part of the device that is called the centricooled unit. And this unit is temperature and gas controlled. So the cells itself never come into contact with the device. They are all processed within a CliniMACS tubing set, so within a closed system. And you see here a CliniMACS tubing set that is installed on the CliniMACS Prodigy device, and all the cells are running within this closed system. So to have a closer look at the tubing set, at the anatomy of the tubing set, we have here an example for a tubing set that is called TS520, and that is used to engineer T cells with a CAR receptor. And what you see here in the upper part of the tubing set, these are the connections for sterile buffer, medium, reagent, and the sample. You see on the bottom of the tubing set are the collection bags for the waste and also for the final cell products. In the middle of the tubing set, you see the peristaltic pump that is responsible for the fluidic movements. And you see here the separation column that is also an integrated part of the tubing set, and where the magnetically labeled cells are separated. Another important component is the gas connection, and that is needed for the cultivation of the cells that take place in the chamber. You see here below. This is as an overview and an introduction to our CliniMACS Prodigy system and the features of the tubing set. And with this, I would like now to hand over to my colleague Philipp, who will give you more insights into the single steps of the car manufacturing protocol and the analytics.
Thank you so much, Martina, for your kind overview. So in the last part of the presentation, I will focus on two different topics. I will introduce in more detail the different manufacturing steps we have to use during CAR T-cell manufacturing. And the second aspect of my presentation will deal with the required analytics, so what kind of quality control and in-process control we have to execute during the manufacturing process, which you see here on the right side of the slide. So as you might have already learned from Michael and Martina, the manufacturing process of CAR T-cell is rather complex and complicated, and many different steps have to be done in the right order that in the end we have a nice product that we can inject into a patient to try to cure him. And those defined blocks here, they have a special name and they are called unit operation. So what is a unit operation? A unit operation is a specific task to manufacturing which has a manufacturing significance. So this involves the usage of a special equipment, special conditions, a special technology to transfer the starting material and the raw material step by step to the final drug product, which can be injected into the patient. And when we think about the CAR T-cell manufacturing process, we usually can distinguish up to 6 different basic unit operations. And these are listed here. We have the T-cell enrichment, which is the first one, where we go and enrich from the starting material the T-cells. The next step is the T-cell activation, which is required to drive them in a state that they can be activated and they are able to proliferate. After this, we have the T-cell engineering, which is required that we can transduce the cell with the lentiviral vector, that the T cells, which we have brought in the culture, express then the chimeric antigen receptor on the cell surface. The next phase is the T cell expansion, where we need to increase the number of our CAR T cells. And then we come to the end, where we have to do 2 steps to furnish our drug product. So first is the harvest of the cultured cell, where we have to formulate them in an excipient, which can be injected into the patient. And the last step is the so-called drug product preparation, which then includes some concentration adjustment to match the specific requirements of the patient or the clinical trial. And then this product can be transported to the patient site, to the local hospital, and the product can be injected to the patient. So this is the entire process, but you see that we have next to these 6 steps, there's something on this side and something on the right side. And this flanking side is not part of the manufacturing process, but it's very important to have this also considered. And this, on one hand, the procuring and the generation of the starting material, which might be a leukapheresis, which is obtained from either a donor or the patient himself. And on the right side, we have to consider also that the drug product has somehow to be transported to the patient side. So there's also some regulation behind how this is done, how long this can take, that the patient in the end receives the cells and is then treated by the responsible physician at the bedside. And in the next slide, we will go step by step through all these important unit operations in more detail. So the first one is the T-cell enrichment. So why do we need to enrich T-cells? Because what you have learned from Michael already is the vector is very expensive. So we do not want to waste them on all the cells. We just want to focus on the important cells, which are mainly the T-cells. So for this, we need to enrich the T-cells. And this can be done on the device which Martina nicely introduced to you, the CliniMACS Prodigy. So here the cells are then loaded into a component of the tubing set, which is called the chamber. In this chamber, we incubate the cells with specific reagents, which then target the T cells. And these reagents come with a magnetic tag. And this allows us to separate the cells on the magnetic unit. There's a column as part of the tubing set where the labeled cells are then retained. And then those cells can be washed off in the intermediate bag, and then they can be further processed. There are different markers that can be used to enrich those cells. There are markers that can enrich all T cells. But this is not very clever to use in manufacturing for the reason that this marker, the pan marker CD3, might interfere with the downstream processing. So most customers go and enrich the T cells by the usage of the CD4 and CD8 reagent, which also nicely enrich both T cell compartments, and this does not interfere with any subsequent processing step. The next unit operation is the T cell activation. Usually this also happens in vivo, in the body, in the human body, when the T cell communicates with a dendritic cell and sees their antigen. And this is a quite complex procedure, so it requires 3 steps to activate the T cell in vivo. The first signal is brought by the interaction of the T cell with the antigen presenting cell. So the antigen presenting cell, which is a dendritic cell, digest bacteria usually and other stuff, and then they present the antigens over MSC complex, and they interact with the T cell via the T cell receptor CD3 complex. And if the T cell sees their antigen, they get a first signal to get activated. The second signal required is also coming from the antigen presenting cell. It's a so-called co-stimulus, and this happens by the interaction of a specific epitope on the T cell, CD28, with other epitopes on the dendritic cell. So this is a second signal, and then we require a third one, which is also provided by the APC, which is then cytokines. So the dendritic cells sense then some danger around, there's danger in the system, and they release cytokines. And these cytokines are then detected by the T cell via their cytokine receptors, and this then activates the T cell, and the T cell start to proliferate. So however, in vitro, this is a little bit different, because we do not want to have just one T cell that is able to proliferate. We want to activate all the T cells. So we require a so-called polyclonal activation, and this is done by the usage of a specific reagent, which in our company is called TransAct. So what is TransAct? TransAct is a polymeric nanomatrix which is conjugated to agonist against these markers here, against CD3 and CD28, which are found on the T cell. And this delivers the first 2 signals. And the third signal, this is then given by the surrounding culture medium, where we have supplemented some cytokines. And this then drives the T cell also in an activated state. They form clusters, and then they start to proliferate. The next step is the so-called T cell engineering step. So what happens in T cell engineering? So we have to think of, we want the T cell in a shape where we have the chimeric antigen receptor on the cell surface. And of course, the T cell doesn't do this on its own account. So we require some help. And this help is used in form of a retrovirus, a lentiviral vector. And first of all, we have to put the genetic information which encodes for the chimeric antigen receptor into a viral particle. And then in our culture, we apply the viral particle to our cells in the culture. And then the genetic information gets transferred to the T cell, it gets transduced and randomly integrated into the T cell genome. And then when this information is expressed, then we have our T cell, which bears then on the cell surface the expressed chimeric antigen receptor. This is considered to be very efficient and reliable, and is also considered to be a safe technology if you have a low vector copy number integrated into the T cell genome. So usually the limit is like you should not have more than 5 vector copies in the cell genome to not set the stability of the genome of the T cell at stake. However, what you might have heard from Michael before is that the production of this vector is very complex. There are high hurdles coming from the regulatory departments and authorities to have this produced in the correct GMP grade, and that all the tests which are required have to be accomplished in the right way, that this can then be released for any batch of a given lentiviral vector. What happens after T cell engineering is, so now we have our CAR T cell already, but the number is too low. So we need to have a higher number to treat the patient. And this is happening in the T cell expansion phase. The T cell expansion phase depends on many different functions. Just want to show you some of them. So usually it depends on the time. So the longer you incubate your cells in the incubator, the higher your cell count will go. It also depends on the culture volume and how rigid you shake the culture. It depends, of course, also on the level of feeding, so how often you replenish the medium, which is indicated by the glucose level here, and many other factors like temperature and pH and gas exchange and so on. The main thing is we want to have, in the end, we want to have two things. First thing is we want to have sufficient cells to treat our patient. So we want to have, regardless of the patient's status, if he is sick or healthy, we want to have enough cells. And we also want to have a good viability in the end, also regardless of the patient status, so that we can treat the patient that we want to treat. So then we already move to the end of the process. So this requires now two steps. But you might wonder, why? Because we have now our cells in the culture, they're happy, we have CAR T cells, we can just go and treat the patient. No, you cannot. The reason is that in the culture right now, we have our cells still surrounding by some raw materials. The raw materials are materials like the cytokines and medium, and these materials, they are fine for production. They have the right grade for production, but this material is not intended to be part of the drug. So these raw materials must not be infused into the patient. So we have to remove them. And we do this by washing, and then we resuspend the cells into a formulation solution, so into a buffer, which is fine that it can be injected into the patient. So a very easy example is sodium chloride solution, which you find in any hospital. This is something that you can infuse into a human being. So this could be an excipient for this. And once we have done this, once we have washed away the medium and cytokines, we have our drug substance. The drug substance bears the active pharmaceutical ingredient into a solution that can be injected into the patient. And the last step that is missing is we have to get our drug product. So we have to have a final step, which is the drug product preparation step. This is a final step. And this final step, is basically we need to adjust the T-cell, the CAR T-cell concentration depending on the required needs for the patient, or depending on what we have applied for. So if we have a clinical trial, there might be specific requirements how the drug dosage has to be furnished. So we create a lot of aliquots, and in our clinical protocol we find requirements how many aliquots we have to produce, for what reason, what should be the dose we have to put into each aliquot, and also in which kind of container format we have to produce those. Important thing is every container which we have produced from our final drug product has a specific intended use. So one is, of course, you need to treat the patient. There might be, depending on the body weight, a defined number of CAR T cells in this aliquot. There might also be a backup aliquot. Then we have, of course, samples required for sample retention, or if something goes wrong in our quality control testing, that we can repeat the test if required. And talking about QC, so quality control, we need to run a full battery of release tests, quality control tests on the final drug product before we can release it to market, before we can release this drug product to be used for treatment of a human being. Okay, this is the end of the first part of my talk. In the second part, I want to come back to the QC test in more detail. Okay, before we dive now into details of quality control, I want to first introduce you what we mean with quality control. So quality control is a quite wide concept. So first of all, we have to think about there's a quality control department. And the quality control department has a quite big responsibility. So it deals with checking or testing that specifications are met. And the main responsibility is they have to ensure that all required tests are carried out and that we do not have any material used into a production process, or that we do not release any product for market or to treat a patient until we have not made sure that this quality is according to specification, that this is fine and we can release it to the patient. But quality control also has a specific meaning here. When we talk about quality control on the CAR T manufacturing area, we often mean the release testing, which we do on the drug product level at the very end of the process. What else is important when we think about quality control is this is a fundamental aspect of the good manufacturing practices, GMP. So every manufacturer has to establish a quality control department, has to establish and maintain this role. And also, you do not need don't have to mix up. So this quality control department must be independent. It must be independent from production to avoid that there is any conflict of interest, that the same staff that does the manufacturing is then also doing the testing of the product and nice procedure, I have done it, I'm the best, and I sign it off, and it goes to the patient. No, it's independent, and the staff, usually, they don't mix with each other. And then, if you think about GMP and have some background, you might know that there's some pillar of GMP. It's not just sufficient that you test for quality in the end. No, you have to make sure that you produce quality. This means you also have to check the quality during your manufacturing process. And this is done by the second aspect here, by in-process control. So in-process control are the tests that are performed during the manufacturing process, during production, and with the purpose to monitor and, if required, also to adjust the process, to ensure that also the process intermediate have the chance to conform according to the required quality specification. And this is very difficult to set up in the process, and usually the level of process control you can execute on a cell-based process, this gains step by step. The more you learn about the process, the more you understand how the process works, the more you can also fine-tune the different aspects of the process parameters, how they impact the final drug, and you can fine-tune your process control. This is very complex, in particular if you think about a living drug like the CAR T cells are. This list shows you an overview of all required methods that are usually have to be executed during a CAR T manufacturing process. And each of those methods, they have a purpose, which I come to in the next couple of slides. So we have an incoming good control, which has the intended purpose that we check the input material, which are the raw material, as well as the starting material, the cellular product that is going to be used in the process to produce our drug product, that they have the right quality. Then we have the purity assay and the density assay, which are set to determine the cellular composition, that we know how many cells we have, what the purity of those cells, of the CAR T cell is during the culture and in the beginning and in the end. And we want to determine the identity of the CAR T cell. This means that we are sure that the T cells, that they bear the CAR construct on the cell surface and not any other cell. Then there's a whole battery of safety testing. So we have a microbiology assay, endotoxin assay, mycoplasma test, vector copy number, and RCL test. And they make sure a couple of things. So the microbiology assay, this should detect if there's any presence of a contamination of a microbe. Then we have the endotoxin assay, which is there to assess the presence in the level of bacterial endotoxin in our sample. The mycoplasma assay is a little bit more complex. This should determine the presence of mycoplasma. And then we have other tests which are PCR based. And one of those is the VCN assay. This determines the number of integrated vector copies per cell. Whereas the RCL assay, the replication competent lentiviral assay, confirms that we do not have by any chance produced an RCL particle in our procedure, which might then set the patient safety at stake. Then we have a potency assay, which determines the functionality of the T cell on a biological level. And the last test is an appearance test. And all these different tests I have introduced you here, of course, you need to plan for those tests. You need to know at which time point during the manufacturing you have to sample, with which kind of container, what is the volume you require, and what are the assays that are required for each sampling time point. And this is defined in a sampling plan. This is an example of a sampling plan, and I just put it here on our exemplary CAR T cell manufacturing process. So sampling is required at different time points in the manufacturing. Usually you do it in the beginning, you do it as an in-process control during the manufacturing, and then you have this whole battery of required testing at the end of the process on the drug product level. So prior to manufacturing, you have a purpose of this because you want to check your input into the process. You want to assess your starting material, you want to assess your raw material, and you also want to make sure that this is not contaminated. This is what we call incoming good control. Then during the process, we want to monitor and ensure that the process performs according to the specification. And at the end, so the end product testing, there we have to run all the release tests to make sure that we have a safe and that we have the product conforming to required quality, safety, and efficacy profile required to treat the patient in the end. So this is very complex and long, a lot of ethics which are then required to release the product to the patient. In our next slide, we will discuss all of these different methods in more detail. And we start with the incoming good control, of course, because it's the first one. So the purpose here is that we control the quality of all the material that goes into the manufacturing process. We can discuss many things here. So what is important to be in mind is that you have to check all the material that goes into it. So it's required for the starting material, it's required for any raw material or ancillary material, as well as for the packaging material that you use in the end to package your drug product. And there are many tests that you can think of, but I keep it simple here. I just want to introduce a visual inspection that you do. So you just check documentation for the starting material, for example. You check, of course, that the documents delivered with the apheresis product is as it should be, that the patient ID is matching maybe the donor ID with the same patient where the cell product is obtained from. You also verify the age of the product. That is the time, means duration time from end of the apheresis procedure until start of the process, that is not too long, because otherwise the product might start to die and you do not want to have clumpy dead cells in your process. And you also want to confirm the absence of aggregates into your product. And when you think about raw material like reagents, they usually come in a glass vial. What you do there, of course, depending on the quality and the available level of documents that comes from the vendor of this product, you verify the documentation, of course, and they verify then, of course, that the reagent is still fine, so it's not expired. And also you want to check that the packaging of the reagent is not damaged. So if it's coming in a glass vial, you check that it's not leaky, that the glass vial is not broken, and so you can happily use it in your process. The next two assays I want to introduce are the purity assay and the identity assay. They both use the same methodology for the analysis, but they have a different purpose, of course. And I start with the purity assay, and here depends on the time point of the process, how you analyze it. And each of the samples you take in the process is assessed for the purity. So on the starting material sample, you assess the purity, and the purpose is here. You want to check that your starting product is fine to be used in the process, so that the starting product meets the requirements for the manufacturing process, that you don't have too many cells, that there are cells at all, that the cells are in a good shape, that they're viable. These are the things that you check in the purity assay. You check this and you want to establish a baseline for the process. Then you do the enrichment, and after the enrichment you take another sample which you assess for purity, and this has a purpose that you want to analyze how well you did do for the enrichment. So what is the yield of your enriched T cells? Do we have enough T cells? Because this also determines then later on for the culture part, how many cells you have to seed in your culture. And this is determined here. Then in the in-process control, you determine your drug purity and the number of cells you have produced and the profile during the expansion. And at the end of the process, of course, you need to determine the number and the purity of your drug substance and the drug product to furnish the right substance, to have enough cells and the right dose into your final drug product container. The identity assay is done not in the beginning, of course, because in the beginning we don't have any CAR T cell. So the first time point where we check for the identity is in the in-process control here. So the purpose here is that we want to determine the identity of the drug substance on the intermediate level. So we want to see what is the transduction efficiency. Do we have 50%? Do we have 20%? Do we have transduced T cells at all? Because if here there's 0%, we can abort the process and start here again, right? So this is a good control time point here to check that we have transduced T cells and that there is enough expression of this construct available. And then, of course, we also determine the identity of the drug product on the drug product level. So here we check the transduction efficiency, and also we want to confirm that the construct is expressed on the right cell. So the CAR should be expressed on T cells and not on monocytes. So we check the identity of the cells. As mentioned, both the purity and the identity assay uses the same technology to assess it, and this is a flow cytometry assay. So what is flow cytometry? It's very simple. In flow cytometry, you stain your cell sample with some specific antibodies. These antibodies, they're conjugated to a tag, which is the fluorochrome, and by this you can apply the labeled cells into a specific instrument, which is called a flow cytometer. And in this instrument, every single cell is analyzed by a laser, and depending on how the cells are labeled, depending on the cell type, the cells emit a fluorescent signal, and this emitted signal can then be quantified and displayed on the screen, and you can then analyze for every cell which phenotype the cell has. An example of such an instrument is the MACS Quant analyzer from our company, Miltenyi Biotec. And the good thing is that we also have predefined ready-to-use cocktails that ease a lot of this complex procedure. So we standardize the sample preparation by using of predefined cocktails we have available for the purity assay as well as for the identity assay. So this enables the user to do the sampling in a couple of, let's say, between 10 minutes. And then we also could use an automated way to acquire the cells and do a fully automated analysis of the data, which eases a lot the pain that usually the customers have when they need to do a manual analysis of the data. Because dating of flow data is a quite subjective thing, and it's a huge benefit if you can have this in an automated and objective manner. And by the help of the CAR-T Express mode, this is very simple and very convenient, and it is an objective way to assess the data and get the results in the end. Now we have a look on all the different assays which are related to the safety. So the first and one of the most important one is the microbiological assay. The idea of this assay is to determine if there's any contamination in the product by a microbial thing like a bacteria or fungi infection or any contamination that you might have in your product. So this is used to determine the absence of a microbial contamination, and it's done on a couple of samples. So we do it on the starting material and to confirm that the starting material is sterile, that there is no contamination. We do it also on the level of the in-process control, as well as, of course, on the level of the drug product. Usually, you require that this last sample is negative, so that this is sterile, there must be no growth. But usually you want to treat the patient rather quick after you've furnished your drug product. And this test takes a couple of days to get the results back. So you cannot wait for 7 days because your cells have a shelf life of maybe like 3 days. So they will be dead after 7 days. And to make it then feasible to have a fresh out procedure, meaning the usage of the cells right after the furnishing of the drug product, we can rely here on the result of the sterility test coming from an in-process control. How is this done? It's a very simple test. So you have your sample and you put your sample into a growth medium. And this growth medium has to promote the growth of any kind of contamination. And if there's a bacterial contamination and they are in a nice sugar medium, they start to proliferate. And then you can see a color change, or you can detect the change of the pH, or you can also see a change in turbidity. You can easily assess this by yourself. So if you have like a bottle of apple juice and you put some yeast inside and put it in an incubator for a couple of days, you find that the yeast clouds the entire juice, and you have some wine in the end and a lot of yeast, of course. And the test works in the same way, and you can assess the result either manually by a visual inspection, so holding the flask against some light and see if you detect some cloudy stuff and turbidity in your sample. Or you can also use available systems from companies that do it automatically. And one example here is the BACT alert system from bioMérieux, which is an automated system where you have a special bottle where you inoculate your sample, you put it in this incubator, and the system automatically does the incubation and also reports back the results, if it's positive or negative. So it's very convenient and easy to use. The next assay is the endotoxin assay. The idea of the endotoxin assay is, of course, to detect the presence of endotoxin in your product. So endotoxin is what is endotoxin? Endotoxin is a class of a pyrogen. So pyrogen is fever-causing, and fever-causing things you do not want to have in your drug product because patients are sick, so you do not want to put some fever on top. But on the other hand, endotoxins are everywhere. So you cannot get zero endotoxin, but there are very, very strict levels of allowed endotoxin contamination. So you have to determine it. And we determine it for sure on the level of the drug product, because this is the end product testing where we have to run all the tests. And we also check for the endotoxin level on the in-process control. How is this done? It's there are many methods for this. So there's a different kind of methods we can use, and all of these methods they involve mixing and incubating of a sample with an enzyme, with a reagent which comes from the horseshoe crab. This is called the Limonus amebocyte lysate reagent, and if there's endotoxin present, you might have either that the gel clot is formed, that there's a color change or a turbidity change, and this can be assessed depending on the assay you're running. And also here, there's a quite clever solution for this from a company called Charles River. It's an EndoSafe system, which can be used for this end. It's a handheld spectrometer, and there's a little cassette. You put your sample in, you apply it to the device, and you can have the result in, let's say, in 20 to 30 minutes, and then you're done. Next assay is a mycoplasma assay. So mycoplasmas also needs to be evaluated. And you might wonder, what Philipp told me before about the sterility test, but why I have to do this test then? Why the heck do we need to have another test? So mycoplasmas are special. So they lack cell wall components. They have many different characteristics, and due to this, the normal cell culture tests do not work. So how can we detect mycoplasma? Is of course also detected at the end product level. We can detect it by different methods. One method employs a culture. So again, a culture, but this culture is not 7 days, it's 1 month. So it's a very long procedure. And this compendium method is quite complex. In the end, you can detect the mycoplasma based on their colony-forming pattern on the agar plate. The second method is called indicator culture. And here you use a dye that binds to the DNA of the mycoplasma, and then you can detect the mycoplasma based on a characteristic formation of resonance pattern. Never did this test. And the last one is a very quick test. It only takes a couple of hours. It's based on a polymerase chain reaction, PCR, and this targets specific genes of the mycoplasm. So if there's a detection of a gene, you amplify this and you get a signal in the end, and by this you know the level, you know that there's a mycoplasma contamination, and you cannot use this, of course, to treat a patient. We stay in the area of PCR tests, and there are 2 tests on this end I need to explain. One is called the vector copy number assay, and the other one is the replication competent lentiviral assay. So VCN, vector copy number, as mentioned before, so we have our vector, we apply it to the T cell, and then the vector infects the T cell, integrates the construct randomly into the T cell genome, and we have CAR-T expression. This happens randomly, and if you have too many copies per T cell, this could put the stability of the genome at stake, and also could cause mutagenesis. And of course, this is something we do not want to have. And for this reason, we have to measure the copy numbers of vector that gets integrated in average per T cell. And there's some guidelines that say this must be below 5. On the other hand, we test also for RCL. So the vectors which are used, they're safe. They're like 3rd or 4th generation of vector. I'm not so much into it. And they're silent. So these, by the way they're constructed, they can infect only once. They can infect the T cell. They deliver their genetic information. But this is not sufficient to produce an infectious particle again. So you cannot, these infected T cells, they cannot produce virus anymore. So it's safe. But there's a little bit of a theoretical risk, like winning in lottery, that you could produce by a rare recombination event, again, a vector that can infect other cells. And to address this risk, we have to run a test to see if we have this or not. And how this is done is on the next slide. So both assays, the RCL and VCN, they use a special kind of PCR, which enables us to quantify the result. So it's a real-time or a digital droplet PCR, and it involves that first we have to isolate the genetic information, so the DNA from the sample, so from our drug product sample or from an intermediate sample. We isolate the DNA, and then we use highly specific components to target different genes. So in the VCN assay, we target a vector-specific gene, which is then also integrated into the genome of the T cell. And this enables us to amplify it, and then we set it into relation with the housekeeping gene of the T cell, and then we can determine the VCN. For the RCL, we try to detect a gene that would be required to lentiviral replication. And of course here, we do not want to have it. So this year, we do not want a result. It should be negative. There should be no amplification detectable. So what do we need for this assay? We need, of course, some isolation reagent to isolate our DNA. We need a couple of chemicals and polymerase to drive the PCR reaction. And of course, we need to have the specific things to target the genes. And conveniently, this is also available from our company to do the VCN assay and the RCL assay. And then you can nicely put these components together. There's also a standard included, which allows you to set up controls and calibration curves, which are required. And then you load this into the PCR device, the thermocycler. You wait for 4 hours, and then you have the result. This brings me to the last two assays I want to introduce. So the next one is the potency assay. The potency assay is a little bit tricky. There's sometimes required among the battery of tests you have to do on the drug product. Depends also in which phase of clinical trial you are, if you're in the beginning or if you're in the commercial phase, if this is required or not. So this is test the biological activity of the drug product and the mode of action. So the theory behind it is very simple. So it's a cell-based assay. This means you take a little bit of your CAR T cells and you mix them in different concentration with their target cells. And then when the CAR T cells sees their target, they don't like each other so much. So what happens, the CAR T cell release cytokines, and this is something you can measure. And one cytokine is interferon gamma, which is also on the slide here. And the other thing is what happens, the CAR T cells, when they see the target, they kill the target. And this is also something that you can measure. So you can measure the response release of the cytokine either by a so-called ELISA method or after binding by some components, also by flow cytometry. Or you can measure also the killing of the cells by other assays. So the theory is very, very easy to understand. The challenging part comes that you also have to compare this reaction with a so-called reference standard. And then it gets very tricky because both reference standard and the drug product have to behave in the very same way. But this is a different story. So the last test I want to introduce you is the appearance test, which is done as mentioned on the drug product level. This usually is a verification of the physical visible state of the drug product, and it involves a visual examination. You have a special background for this, inspection background, and you check that you have the right label on the drug product. It's nicely done and sticks to the back. You check if there are any signs of aggregates in your drug product, because aggregates is something that you do not want to have in your formulated drug product, because if those get infused into a patient, the patient might suffer from thrombosis. It's not so healthy and you don't want to have that. You also check that the color is correct, so that the drug product has the expected color, the expected turbidity, and you also then check the container closure, that it's not leaky. You check the integrity of the bag, and once you're happy, we also are happy, and that we can infuse this product then after all the other tests perform up to a specification to the patient. Okay, with this, I'm at the end of our lecture, and I want to thank you very much for your kind attention, and I hope you learned something new, and if you have any question, you're welcome to come back to us at any time. So thank you.